Okay, thanks very much for this opportunity to talk about the Australian Imaging Service. So you may recognize from here that unlike a lot of the talks, the Australian Imaging Service has a biomedical focus. Still has all types of images in it, but if a lot of what I'm talking about is a bit unfamiliar, that could be why. So the Australian Imaging Service is a platform for collaborative imaging research, which encompasses the ingestion of data from clinical sites, secure data management, access controls, and the identification of sensitive data, the ability to view, annotate, and analyze images through your browser, and also integrated analysis pipelines, both a curated suite of pipelines managed by the AAS core team, and the ability for the community to contribute pipelines, whichever one can use. And so it aims to increase reproducibility and adoption of trusted analysis methods across the network. So the AAS is organized as a federation. Currently 12 institutions running their own AAS instance. So you can see each of these nodes are connected to the instruments via these edge devices, which typically sit on the network where the instrument is, so potentially at hospitals or research facilities. And these upload the data from the instrument up to the AAS node, which contains the data repository and also the integrated analysis tools. So plan for the future is this sort of one-stop portal onto the AAS, so users can come to that portal and then find data anywhere across the network. And underpinning it all is the sort of common code repository, which is up at our AAS GitHub organization, which sort of ties it all together and is open source for anyone to use. And so one of the big benefits of this federated approach is it allows each institution to control its own AAS nodes and use whatever institutional computational resources are available to them, and also adhere to the local policies around data management. But looking into the structure of each of the nodes, so the foundation is this data management features, so which enable like secure collaboration and client-side visualization. But then we also have graphical analysis, virtual desktops, Jupyter notebooks, which enable sort of bespoke analysis and exploration of the data without needing to download it. And then the automated pipelines, which are particularly useful for quality control, file conversion, some more extensive pre-processing all the way up to bulk analysis of large data sets. And then in the final feature set is the machine learning, which includes AI-assisted segmentation, which can then be fed into training, and federated learning. So looking at the technology stack that behind all this, you'll see that in the core, the center of it is the XNAT data repository platform. So this is sort of analogous to Amero, but with a biomedical imaging focus. On the acquisition side, we have the Medical Imaging Resource Center Clinical Trials Processor or the XNAT ingest package that we've developed, which do sort of initial sorting of the data, so it goes into the right project on XNAT, and also de-identifies and securely transfers over the internet to the typically cloud-based repository. So if users want to access the data, they use the OpenID supplied by the Australian Access Federation for authentication and authorization. And then on the right-hand side, we have our automated pipelines, which run within Docker containers, a Jupyter Hub, Neurodesk for the virtual desktops, and then machine learning through MonEye and NVIDIA Flare for the federated learning. And so you'll notice here that all this sits within a Kubernetes cluster, For those not familiar, Kubernetes is the technology which Google developed to run Google. So it's highly scalable, powerful framework. And it's really nice for this project, and really what makes it feasible is that we can come up with these Helm templates, which are sort of like a cookie cutter approach that we can then produce these multiple nodes at different institutions, but maintain the same code base, and then use customized for any minor adjustments that need to be made for each of the sites. So just have a deeper look at the data management features. So as I said, yeah, at its core, it's we use XNAT for the data management. This has an extensive REST API, which is really useful for scripting and building in other functionality. It's secure, searchable, and it has project-based user controls, which can be controlled by the project CI. So once the project is set up, the sort of user access to the data is fully in the hands of the CI. And another nice feature is this XSync plugin, which enables us to transfer data from one XNAT to another. So in this way, we can sort of push data around the network for national projects. And so this is a feature that's currently in development, but for participants that might access multiple nodes, or might visit multiple nodes, or have the data in other repositories such as REDCap or Australian Biocommons. We're working with the Australian Access Federation to mint global unique IDs, which are sort of cryptographic hashes generated from identifiable information, such as the name, address, and date of birth. So this is sort of one-way hashes, which means that you go from the identifiable information to the hash, but you can't come the other way. So you can use this to link the data across the different nodes, other platforms. So for the graphical analysis, as I said, we use the Jupyter Hub here, and through the XNAT's Jupyter Hub plugin, we can list a number of notebooks, have of pre-configured notebooks, including this specialized Neurodesk one, which launches this virtual desktop for analysis. I'll explain a bit more next slide. So this was recently just published in Nature Methods. It's actually a sister project of the AIS. It's funded out of the same funding pool. And it provides like a Linux-based virtual desktop with all the image analysis tools readily accessible. And it does this by quite a neat system where all the tools are installed in Singularity containers, which are stored within the CERN virtual machine file system drive in the cloud, and then pulled down to the virtual machine, which can be a virtual desktop, which can either be running in AIS or on a user's laptop, or even on HPC, it can be set up as well. So you can have the identical tools available on whichever platform the user wants to use. So then just onto the automated pipelines. So when we sat down the initial remit for this was to produce some pipeline suites to run analysis methods on the imaging data. But we soon realized that in order to maintain like a large suite of analysis methods for all the wide range of data that exists within the Australian Imaging Service would be feasible. So what we wanted to do, and what we did instead, was to create a framework, a deployment framework, which makes it easy for users to contribute their own pipelines, which we can then look to migrate into the curated side of the library. So we did this using GitHub, where we have simple YAML-based recipes to build these containerized workflows, which then get automatically pushed out to each of the nodes in the federation. So the the anatomy of these containerized images, where you have a simple workflow shell command which doesn't know anything about XNAT, but then the pipeline to app package will then install itself within it and then handle the source node to pull the data from XNAT and this over to the workflow, and then the sync data will put it back into XNAT again. So this package is able to sort of keep all the sort of boilerplate or away from the workflow, so the imaging scientist can just focus on the analysis that has to be performed and not any of the integration with XNAT. Finally, just onto the machine learning. So the XNAT has been, we're working on this integration with Monet, which is like the leading medical imaging AI project out there, and other major players. And so using XNAT's OHF viewer, you're able to, radiologists are able to go through and mark out different, segment out different parts of the images to train the machine learning algorithms. And also, this also includes AI-assisted segmentation, which makes that process a lot easier. And then finally, we also have the federated learning feature, which is for data which is too sensitive to upgrade, to really leave that the site where it's acquired. So maybe we have, in one case, we have full body photography data, which needs to stay on site. So what you can do instead is you can perform model training on each of the different sites in the node, in each different sites, and then actually share the model parameters to update a centralized model instead of having to share the data to a centralized node. And so, yeah, this is quite a, there's quite a lot of interest in this, but it's quite difficult to set up a framework to do this. So having this already set up for researchers to come along and use, we're hoping will facilitate quite a few projects. So I'd just like to thank everyone involved, in particular Ryan Sullivan, who's the lead of the project and the real driving force behind it, who would have loved to be here for this, but just had his second child recently, and all the funders, including the National Imaging Facility, AIDSC, Medical Research Future Fund, European Commission, and all the four institutional partners.